All right, everybody, we're gonna see how fast we can fire through this. Just because a bunch of you were telling me like we got like they're getting long, and I need stuff to cover for midterms. So, yeah, here we go. 2.5: Investigating ecosystems, practical work. I did not cover the detail the book does about measuring. I just didn't. Overall, this chapter is worth reading for your IA ideas. So you're not going to hit this up until we're done with chapter 5 probably, and you're going into your IA, which is probably like right off the bat senior year, but it's worth giving a read. Studying ecosystems, IB animal experimental policy. So basically, you can't really experiment on animals if you're IA, and IB you just can't. Nothing's going to harm them. So people included. So can't have pain, undue stress, or damage that will then lead to death, and you definitely can't have death either. So can't have any sort of drug intake. Nope. Dietary change that's not easily tolerated. And be careful there because they're the ones who dictate what's easily tolerated, not you, even if it's your own body. Um, should think about replace animal by a sim, cells, or plants. Reduce the number of animals. Humans need written permission and no blood-borne pathogens. So basically, good luck doing any sort of research on any sort of animal. Just don't. I mean, they're looking for you to use sims and stuff. Like they're looking for anything other than on animals. Techniques for data collection. Where to collect quadrants, transects are good ones to go with. Yep. Let me move this for a second. Yep, over there. Sweet. So what to measure? Abiotic factors, stuff that does not living, marine and freshwater and terrestrial. You can measure abiotic factors in all of those. Biotic factors, you can measure biomass plus productivity, catching small motile animals. That gets a little bit, it's more like catching bugs, really. You're not out there catching mice or squirrels or any of that. I can tell you that. Maybe catch geese like off of, on the field. Measuring abundance. Define Lincoln index for me. So, yep, go for it. Knock yourself out. Figure of speech. And then define Simpson diversity index. I'll move that up a little bit. So, where to collect the data? Quadrants. Copy data table from page 127. You can tell if your quadrant is big enough because eventually your numbers will stabilize. So when you copy the data table from page 127, it's going to look something like this. Yep, 127. I think page 127 is right here. Yep, so I'll make that big. Yep. Copy down the data table right there. Beautiful. All right. Now, exit full screen. Yep, get the notes back up there and make that big. Good. All right. How to replace quadrants randomly or systematically. So, um, first, map the study area. This for randomly. Second, draw a grid that covers the whole area. Third, number each square. Fourth, pick a random number to measure. So, that's how they want you to do it a little bit randomly. It doesn't seem too random to me, but it's you know, you're mapping the study area that you choose and picking a random number to measure. So, stratified random sampling used when when differences are obvious between samples. Two sets of data must be taken, at least one in each area. Um, First, draw a grid for each area. Second, number each square. Third, randomly select a number from each obviously different areas. Transects might use this in an area that transitions. Shore to mainland is one of them. Let's see if I can get that up there. Yeah, I'll move me over here. Good. Valley to a mountain, farm field to a forest is a real common one. That's the one that we would have issues with here at school. Field to a forest. Need three, preferably five transect datas to be valid. So, two types of transects. Line transect equals a line, but put towards the gradient and species touching the line are recorded. Belt transect equals area of chosen width between two parallel lines, usually zero point 0.5 to 1 meter wide. Continuous transect equals whole line or belt sampled. Interrupted transect equals samples taken at point in the line or belt. What to measure? Measuring abiotic components. No matter on land, salt or freshwater, it's going to have abiotic factors. Yep, you got things that aren't living. Should be able to describe them, should be able to measure where they are and when. Why is dissolved oxygen important in marine ecosystems? Look that up for me if you can. I'd love to know. Define salinity. All right. Salinity can be determined with the electrical conductivity or density. What's the pH of ocean water? Is it acidic or basic? What would affect the pH of the ocean? Dumping batteries? I don't know. Probably more like with plastics, possibly. Oh, metal erosion. All right, heavy metals. Wonder what that would do. Um, what is the ocean's role in the carbon cycle as well? Temperature. All right. Many marine organisms are ectothermic. Define ectothermic. Low temps equal low metabolic rate. So, water temperature controls where sea creatures live or no longer live. Fish are very temperature dependent. Other sea creatures as well. You'll see them, there's a line in the water, and it'll be like, okay, warm water here, cold water here. And if they're cold water, they will not cross that line, even if a shark's chasing them. They're like, nope, not going to happen. Think of someone in your grade that has a high metabolic rate. So what reason for that? Think of someone that is like constantly moving. High energy, tends to be pretty skinny, can eat like a gallon of ice cream and doesn't gain a pound. All right. Dissolved oxygen. All right. Affected by temperatures equals higher temperature means less dissolved oxygen. So the more ocean water, the more we have global warming with sea temperatures rising, less dissolved oxygen we have in our oceans. Pollution, more pollution means less dissolved oxygen as well. Kind of goes hand in hand with the warming. Waves, areas that, with waves tend to have more dissolved oxygen. Coral reefs, rocky shores, stuff that's really going to get that water turning is going to get dissolved oxygen into it. Freshwater ecosystems, so abiotic factors equal turbidity, flow, velocity, pH, temperature, dissolved oxygen. Turbidity, high turbidity equals cloudiness. So if you got like something that's like orange juice, it's cloudy. You can't see through it. You got something that's more like the water out of your, water fountain. Yeah, it's going to be more clear, low turbidity. Name a high and low turbidity liquid. Oh gosh, I just did that for you there with the OJ and the water. So more turbid equals less sunlight equals less photosynthesis. That's swamp water. Not a lot of photosynthesis going on with it. Flow velocity. Move this down to right there. Okay. Equals speed water is moving. Determines which species can live where. Some need more, others less. So if the water's moving too fast, it will rip. Good luck. With fish swimming upstream. All right. If the water is too low, other species have other problems. Like good luck if you're a filter feeder, like a clam, like an oyster. So time. Spring snowmelt flows fast. Summer drought flows slow. Depth. Surface flows slower, deeper flows faster. Position. Inside the river bend flows slower, outside the river bend flows faster. So, yep. pH. Range, but generally slightly acidic to slightly basic. Depends on soil, rock, vegetation, and pollution. So, terrestrial ecosystems: air temperature, light intensity, wind speed, soil texture, slope, soil moisture, drainage, and mineral content. So, air temperature, light intensity, wind, rain, rainfall, soil. So, these are all abiotic factors you got to deal with. Soil, there's a lot too that people don't realize. People think it's just soil is soil. Not really. There's good soil, there's bad soil. Good dirt and bad dirt. Just make sure they don't got good dirt on you. So, all right, now texture: gravel, sand, silt, clay. You need like a mixture of all of those for different reasons. Just for holding on to water, but also letting air on in the flow of water. Soil moisture is important because obviously dry soil, you're not growing crap there. So you got to have organic content in it. Organic content is stuff that's breaking down. You want a soil that's dark and rich. If it's not dark and rich, the organic content is pretty low with it. Organic content is what carries the nutrients. It holds on to water. Reduces compaction, which is just like soil that's just been beat down into layers. Crusting. It increases infiltration of water, so it lets water in, but if it's too much water, it just lets it go right out, which is a good thing. So mineral content and pH is important as well, and that also works in with soil. Measuring biotic living components of the system. You need to observe and question measuring biomass and productivity. Plant biomass measuring is simple but destructive. Best to only include plant above ground. Don't include the plant below ground. No one's interested in the roots, really. Primary productivity. In aquatic habitat, book describes a way with two bottles. In terrestrial, book describes using square patches. So, you know, secondary productivity. Problem is, it requires an animal, human, and measuring poop. Dude, no. You don't want to get in with that crap. So, no secondary productivity studies going on in my class. Catching small motile animals. Tough because they move. Yeah, mice, you ain't going to catch them. All right. Insects, you got a shot at. NIB, no animal can be stressed or killed during investigation or have anything done to them, which will lead to them coming down with a bad case of dead. So, mostly insects, terrestrial ecosystem, pitfall traps. Great for insects that can't fly. Use decaying meat or sweets. Check every 6 hours, not longer than 24. So, one of those things you got to put up one day and then take down the next. Sweep nets, swept through grasses, use at different heights, put insects in large clear containers. Tree beating, good for finding bugs in tree branches. Tray underneath branch, tap the branch. So it's not like you're beating the tree with a baseball bat like it's a pinata, but you definitely want to like put a tray underneath some branches and then just put a rope around the branch and just shake it. Light behind the white sheet at night is a way to do it too, to see like what. So it's almost like creating like a spotlight to see. Problem is, if you're doing that at school, you're on school property after hours, you won't be dealing with me. All right. Image search and draw a pooter jar for me. So yeah. Aquatic ecosystems. Kick sampling. All right. Good for stream invertebrates. That's probably sampling. There we go. Nets are very useful, or sieves as well, in the aquatic ecosystem. Turning stones over is good. So, keys, biological keys, kind of like a visual cheat sheet, shows you like what you're identifying. There's two types: the diagrammatic diatonomous spider key. I'm not, you know, and then there's a statement key. More exact because words are more exact than pictures. So, yeah. Look at page 138 and draw an organism from the diagrammatic key. Measuring abundance, plants. So let's draw, let me show you page 138 here. Here we go. This is page 138 right here. So they're going to want you to draw one of these, I believe. So, yep. Yep, just draw an organism. You don't have to draw the table, just an organism. So, should probably highlight that. And we'll make that green. Good. Measuring abundance, plants. Density, mean, which is average number of plants per meter squared. So frequency is the number of the total quadrants species were found in. Percentage cover, more useful with plants than individual numbers. Estimate the amount of in a quadrant covered by plant. Copy data table, figures 2.5.17 on page 139. So, yeah, let me show that one. This is the data table right here on page 139. Copy that one down for me if you could. All right. Lincoln index, capture, mark, release, and recapture. Used for fast animals. First, establish a study area. Got to do that. Second, capture a sample of the population. Third, mark each organism. Fourth, release the organism. Fifth, capture another sample. See what percent is recaptured. At least 10% for it to be accurate. Copy the equation from page 140. Yep. All right. So let's go page 140. Yep. Equation right there. So copy that down, please. Simpson diversity index, used more for vegetation, but can be for animal. Higher the number equals more diverse. So it's good for comparing ecosystems, see if the ecosystem is changing. For a much better explanation and spelling, all right, look at page 41, 141 in the book. So, yep, that's pretty much where we ended off. So, yep. All right, peeps. Hopefully that helps. I know this has been a fast one. So, later.